So when a peptide or protein is first discovered and isolated and purified, one of the first pieces of information that we want to get about that protein is its amino acid composition and its amino acid sequence. And so the method for doing this, for sequencing of a protein, was developed by Frederick Sanger all the way back in the 1950s. And so I'm going to go through those steps, and then we'll do a practice problem in another video to get an idea of how you can use sequencing data to actually piece together the amino acid sequence of a molecule. So usually the first step when you're trying to figure out what the order of amino acids in a protein is to figure out what amino acids are present, period. And so the way that we do that is by peptide hydrolysis. And that will allow us to get the amino acid composition. So it won't tell us the order, but it will tell us what amino acids are present and what their relative amounts are. Now, typically the way we do this is in an acid-catalyzed fashion. So it's basically like taking the protein and dissolving it in something like 6 molar HCl. And what that's going to do is it's going to break, it's going to hydrolyze all of the peptide bonds in that molecule. So they're all going to break apart. So what we're looking at here, if we want to look at the chemistry of that, so if this is a peptide bond, so I'll just use these little squiggly lines here to represent the rest of the protein. But we're looking right here at the peptide bond. So that's the amide linkage linking two amino acids together. So in the presence of acid, we first get a protonation step where we protonate the oxygen on that carbonyl, making it positively charged overall. And what that's going to do is that's going to facilitate a water molecule coming in. So here's my water molecule with its 2 lone pairs of electrons. That's going to facilitate the hydrolysis reaction by making it even easier for that lone pair of electrons to come in here, attack that carbonyl carbon, and then kick these electrons back up to that oxygen. So if we draw that, we're now looking at something like this. We've still got that hydrogen on there from when we first protonated, but we don't have that positive charge anymore. So there's the rest of the molecule. Here's where our water molecule is now attached. There's the lone pair. It's now a positively charged oxygen. And then here is where that amide bond was. And remember that nitrogen is also attached to the rest of the protein. So now we can have a proton transfer to actually move that proton off of the water. So we'll have the lone pair of electrons from this nitrogen atom right here. And that will allow us to give this pair of electrons to the oxygen and get rid of that positive charge. So if we draw that structure now, so we've still got this hydroxyl group up here. We've got a hydroxyl group here now because we got rid of that proton by transferring it over here. And now this nitrogen is positively charged because we did that proton transfer. So now the electrons can fall back down to reform the carbonyl, which will allow us to break this amide bond. So remember, this was the bond here that we were trying to break in the first place. So when these electrons fall back down, we can now break this amide bond, facilitated by giving that lone pair of electrons back to the nitrogen to get rid of that positive charge. So there's my arrow from the last page. And so now we've got the carboxylic acid here. And we've got the other amino acid that we've now broken off, whatever that may be. And now that oxygen is again positively charged. Oops, why is that not drawing? So now we can do one last proton abstraction to get rid of that positive charge. And now we've got, there's the carboxylic acid of one amino acid, and there's the amino terminus of the other one, and of course, at physiological pH, that hydrogen right there, that proton, is going to ionize. So then what we can do is, once we've completely hydrolyzed the protein and we've broken all the peptide bonds, we now have this mixture of amino acids that we can now separate by high performance liquid chromatography. So you can see an example of an HPLC trace. Oh, I'm writing in red now, sorry about that. Of an HPLC trace in your textbook on page 102, figure 3-31B. And you can see that each amino acid, when run over this HPLC column, annoying, that's not an HPLC trace, that's a mass spec. I guess they got rid of the HPLC trace, so I'm going to have to show you my own. So hold on a second, and I will put my own picture in. Okay, here's one. This is a crude picture, yet functional. So running this over a HPLC column that has typically a hydrophobic column resin, so we're doing hydrophobic interaction chromatography. Along the x-axis, you see that we have minutes, and along the y-axis, they've got millivolts, which is basically just the signal intensity. So sometimes you'll see like absorbance units or something like that, fluorescence units along the y-axis. But you can see that each particular amino acid in this particular sample has its own peak characteristic, so its own elution time that it comes off the column. And then the amount, or the height of that peak, how much signal it's giving, is proportional to how much of that particular amino acid there is. So from that information, you can get the relative amounts of all the amino acids present. Now, a tweak on this experiment is, before doing the peptide hydrolysis, you label the protein with a molecule called FDNB. That stands for 1,2,4-dinitro-benzene. And what it does is it tags, oops, I can't spell, it tags the N-terminal residue. So whatever amino acid is first in the amino acid sequence. So the way that works is FDNB has a structure that looks like this. So you know it's 2-nitro, and there's our fluorine. So if you react that with a free amine, such as on the N-terminus of an amino acid, so I'll put R1 to represent the first amino acid in the sequence, and then it just goes on. So you get a reaction right here. And this molecule, the FDNB, is optically active. And so you do have to keep in mind, though, that anywhere that there's a free amine like this, you can get labeling with FDNB. So you do have to consider side chains of amino acids that might be reactive, but you can typically figure that out based on the results that you're getting. So in the next video, we'll look more specifically then at how you get the order of the rest of the amino acids. So this video was just how do you figure out which amino acids are present, what their relative amounts are, and which one is first.